Tonight, on the Dreamy Physicist, a strand of life hums quietly inside you. It has no voice, no sound, and yet it has always been singing. Hi there. I'm so glad you're here tonight. You've made it to this quiet place where science becomes a soft lullaby. So let your shoulders drop. Let your breath deepen. You don't need to solve anything now. Just listen, gently, as we drift together into the spiral script of your becoming. Tonight, you lie still beneath your blanket of stars, and your breath rises like the gentle tide of a sleeping sea. All around you, the world softens, quiets, folds inward, and within you, something ancient stirs. Not a thought, not a dream, but something far older, the silent rhythm of your cells. Your body hums with a music you cannot hear, but that you have always known, a hidden song, curled like ribbons inside every living thing. It has no melody, no voice, and yet it is the blueprint of your hands, the tone of your laughter, the spark behind your eyes. This is your DNA, not just a molecule, but a library of memory written in code, a whisper passed down across a billion years. You are not alone in the dark tonight. You are cradled by instructions older than fire, carried like a candle flame from one generation to the next. Imagine this code as a long, spiraling staircase, a double helix of four simple letters, A, T, C, and G, just four. And yet, from them springs the soft blush of your skin, the steady pump of your heart, the dreams you carry in your sleep. They curl and twist, fold and open, shifting like origami to become the cells that make you. Each one of the trillions of cells in your body contains this entire message, a complete copy of you. And though you cannot feel it, these letters are at work, even now, guiding the renewal of your skin, the repair of your bones, the rhythm of your breath. It's like a quiet script being read aloud in every corner of your body, not with words, but with chemistry, with shape, and folding, with dance. You inherited this whisper from your parents, and they from theirs, in an unbroken thread that traces back through centuries of love and chance, of mistakes and miracles. You are part stardust, part memory, part cosmic poetry. Close your eyes if you haven't already. Imagine that spiral of code floating gently before you. Not cold, not sterile, but glowing softly like a strand of moonlit silk drifting in a still pond. It doesn't tell you who you are, but it offers a map, a suggestion, a gentle pull in certain directions. Your genes say, your eyes might be this color, your hair may curl like this, your body might respond to the world in this way. But still, you are more than your code. You are the way your fingers trace music in the air. You are the scent that reminds you of home. You are the laughter that surprises even yourself. You are the places your DNA never predicted, the parts shaped by love, by story, by the choices you make in quiet moments. And yet, we return to the spiral, because it is where the story begins. It is strange to think that all of this, your life, your thoughts, your unique way of being, can be traced back to a silent molecule drifting in the dark of your earliest cell. And stranger still, that we can read it now, letter by letter, instruction by instruction, we can peer into this sacred thread. It took thousands of years of questions, of myths and medicine, of wondering why children look like their parents, why some fall ill and others do not. It took monks in gardens, scientists in glowing labs, countless dreams and failures. And finally, we learned to look at the spiral itself, to read the language of life, to find the patterns, to know where the instructions live. But now, something even more remarkable is beginning. We are no longer just listeners of the code. We are beginning to speak back, to write, to edit, with new tools, delicate and sharp as moonlight. We are learning to touch the spiral, to trim it, shift it, remove a faulty step, replace a broken phrase. We stand at the edge of something profound. For the first time, we may choose not only to observe what is written, but to change it. Imagine a painter standing before the canvas of life, brush in hand, deciding where to add light, where to soften a shadow, or a poet rewriting a single line in a poem that echoes across time. That is the power we now hold, and it is as beautiful as it is terrifying. What if we could prevent illness before it ever begins? What if we could gently correct a flaw in the code and set a child's future on a new path? What if we could write health, intelligence, even beauty into the script of a life before the first breath is drawn? And what if, in our eagerness to help, we go too far? What if we forget that the poem is already beautiful, that some of its flaws are also its music? These are the questions that float around us now, like dust in a quiet room, not demanding answers, but waiting to be felt. So tonight, you rest beside this unfolding future. You lie here, as you are, unmodified, unique, unfinished, and all around you. Science tiptoes toward the spiral, curious and careful, holding its breath. Somewhere, in a petri dish or beneath a microscope, a new hand reaches toward the spiral. You whisper, returning the call. And from this moment on, we are not just listeners. We are editors. But before we begin to write, before we lift the brush or take the scalpel, we must understand the language we hold. So let's begin with the basics, with the simple, elegant alphabet from which you are made. Let's step softly now into the next chapter where the code begins to breathe. You are still resting, your body gently sinking into the quiet. And somewhere deep inside you, the spiral continues its soft whisper. But tonight, let's look closer. Not just at what your DNA is, but how it moves, how it folds, unfolds, opens its arms, and closes its windows, like a living house that breathes along with you. You may think of your DNA as a blueprint, a static instruction manual tucked away in the vault of every cell. But the truth is more delicate, more alive. It is not a dusty scroll. It is a dancer wrapped tightly in spirals, but ready to stretch, sway, respond to the music of your life. Because the code, it breathes. Let your imagination drift now into the warm interior of a single cell, tiny, invisible, yet alive with movement. There in the center, nestled like a pearl, sits the nucleus, holding your DNA in its arms. The spiral, too long to lie straight, wraps again and again around spool like proteins called histones, like thread coiled around fingers. It is not chaos. It is choreography. Sometimes the code is tucked in tightly, its pages sealed, unread. Other times, a portion loosens. An invitation is made. A cell reaches out, opens a page, and reads single line. It doesn't read all of it. No cell ever does. Because though every cell holds the full book of you, each cell reads only the chapters it needs. A skin cell reads the instructions for skin. A neuron opens only the pages about thoughts and fire. A liver cell hums its own song and leaves the rest in silence. This selective reading is part of the wonder. And how does the cell know what to read? Ah, that's where the breathing begins. Imagine switches, tiny, invisible light switches, sitting beside the lines of genetic text. Some are turned on, others remain off, perhaps forever. These switches are part of the epigenome, a soft layer resting just above the code itself. EPI, meaning above, like a filter or a score on top of a melody. It is not your DNA that changes, but the way your cells read it. These switches are flipped by the world around you, by temperature, by light, by stress and love, by the food you eat, even by the memories your body holds. A mother's voice can turn a gene on in the womb. A trauma can silence one. Your experiences speak softly to your code, and the code listens. Can you feel it? Your DNA is not a frozen script. It is a living story, one that you co-author without even knowing. Every breath you take, every choice you make, leaves a fingerprint on the way your body interprets its blueprint. And the most elegant part? This dance is reversible. These epigenetic marks, these gentle switches, can be turned on and off again. They are not scars. They are suggestions. If your body once needed to be alert, a switch flips. But if peace returns, the switch may quiet. You are not trapped by your genes. You are in conversation with them. Like a violin tuned to the room it's played in, your body responds to its context, and so does the code within you. Now, imagine this. Two identical twins, born of the same DNA, but raised in different homes, under different skies. One spends years in stillness and solitude, the other in laughter and bright chaos. Years later, their faces may still mirror one another, but their bodies may not respond the same. One may carry a risk for disease that the other does not. Why? Because the switches have changed. Not the book, but the reading of it. And isn't that comforting, in a way? That your future is not sealed in a spiral. That your life writes notes in the margins of your biology. This breathing code, this quiet adaptability, is what allows life to survive. In a forest, a tree may change its gene expression in response to drought. A bear may shift its metabolism as winter nears. A child's body learns to fight a new illness, flipping on defenses, written long ago, but never before needed. The code is always listening, always ready, always breathing. So now, as you breathe slowly in and out, know that you are part of this ongoing dialogue. Every cell in you is reading, folding, responding. You are not static. You are symphonic. And this opens a new kind of question, doesn't it? If the code can change how it's read, can we too learn to guide that reading? Can we flip the switches ourselves? Scientists are beginning to try. They're learning to understand the epigenome, this soft layer of light and shadow that paints your DNA with possibility. They dream of turning off genes that cause inherited diseases and turning on the ones that protect and heal. They imagine personalized medicine, not just based on your DNA, but on how your life has shaped its breath. It is still a beginning, a soft footstep into the forest of potential. But the trees are rustling, the code is listening, and the future may hold a new way of healing, not by replacing your blueprint, but by learning its rhythm, its pauses, its breathing. So tonight, as you sink even deeper into rest, know that inside you, the code is not asleep. It is stretching gently in the dark. It is opening the right pages. It is keeping you whole. And just beyond this gentle silence, a new tool is arriving. A way to go beyond reading, beyond switching. A way to reach into the very letters themselves. But to do that, we'll need something precise, something sharp and light as starlight. We'll need a scalpel made of song. We'll need CRISPR. You're still adrift in the calm, wrapped in the stillness of your breath, where nothing is urgent, and even your thoughts have softened into the rhythm of a tide coming and going. Inside you, the spiral of life remains, coiled in quiet beauty, still breathing, still folding, still responding to the world, without ever making a sound. And tonight, something extraordinary approaches that spiral, not with noise, not with force, but with light in its fingers and memory in its hands. This is CRISPR. Say it softly. It is not a harsh word. It floats like mist, like frost on glass. CRISPR is not a tool forged by humans. It is something we borrowed, something nature discovered first, long before we had names for anything. The story begins not in us, but in the smallest creatures, bacteria, alone and unprotected in a sea of viruses. You see, viruses don't just attack us. They've been attacking bacteria for billions of years. And over time, bacteria learned a trick. When a virus invaded, they captured a tiny piece of it, a fragment of its code, and saved it deep within their own DNA. A memory, signature, a warning. And the next time that virus returned, the bacterium would recognize it instantly. It would send out a guardian, an enzyme guided by that viral memory, to seek out the matching intruder and cut it. That enzyme was called CAs9, and the memory it carried, a strand of RNA, became its guide. That defense system is what we now call CRISPR, clustered, regularly interspaced, short palindromic repeats. Yes, it's a mouthful, but all you need to hold on to is the idea of recognition, of matching, of memory. CRISPR is a search tool, a pair of eyes that knows what to look for. CAs9 is the scalpel, the hand that cuts once the match is found. Together, they move through the spiral of DNA like a dancer tracing a path they've rehearsed 1,000 times. When CRISPR enters a cell, it carries a copy of the target code, the part of the DNA we want to find. It doesn't guess. It searches line by line, letter by letter, until it finds the perfect match. And then it snips. One tiny, careful cut. This is not destruction. It is possibility. Because once that cut is made, the cell tries to fix itself. It patches the wound. It edits the line. And we can guide that repair by offering a new version of the sentence. Corrected word. A different ending. In this way, CRISPR becomes not a destroyer, but a rewriter. Imagine editing a single line in a book the body reads every day. One small change, and suddenly a broken protein works. A silent gene speaks. Faulty switch is set right again. This isn't just theory. It's happening. In the last few years, scientists have used CRISPR to repair genetic diseases in animals, to silence harmful genes, to awaken healing ones. In people, it's already being tested. In sickle cell disease, inherited mutations cause red blood cells to twist into painful, damaging shapes. With CRISPR, scientists have edited those cells, turning back on a gene for fetal hemoglobin that had long since gone quiet. And the cells began to heal. It's not a fantasy. It's already in motion. But like every tool of great precision, CRISPR is only as responsible as the hands that hold it. Because while it can correct, it can also make mistakes, off-target cuts, unintended changes. The spiral is vast, and even with the best maps, we sometimes stumble. That's why science moves slowly, not out of fear, but out of respect. Because the power to edit life is not a tool we wield, it is a relationship we enter. You are not just cutting DNA, you're shaping futures. And with each change, you send a ripple forward through time. Now imagine this. What if CRISPR is used not just to heal someone who is ill, but to alter an embryo, to make changes that pass on from parent to child, through every cell, through every generation? What if you could choose your child's resistance to disease, or their eye color, or their height, or their intelligence? This is where the silence deepens, because healing a body that suffers feels noble, but choosing traits in the unborn that begins to feel like something else. A door we are just beginning to open, and once it opens, we may not be able to close it again. CRISPR is no longer just a tool for treatment. It becomes a tool for design. And now we are not simply responding to nature. We are attempting to author it. Can you feel the tension? The hush in the room, where science meets philosophy. Because the questions are not technical now. They are moral. They are human. What do we change? What do we leave untouched? Who decides what a better version of life looks like? And how do we protect what is beautiful about imperfection? You don't need to answer these questions tonight. Just feel their weight, like stones on a riverbed, silent, but shaping the current. CRISPR offers a future where fewer people suffer from inherited illness, where bodies can heal themselves before ever knowing pain. But it also offers a future where choices are made before a life has a voice. And those choices may shape the kind of world we wake into. So tonight, as you lie in the soft hush of wonder, picture the spiral, and the scalpel of light moving through it, not to destroy, not to control, but to understand, to guide, to ask, always, not just what we can do, but what we should. And as the spiral turns softly onward, we step closer now to the edge of intention, to the quiet, trembling line between healing and enhancing. You are quiet now, still and weightless, drifting in the gentle dark. Your body is resting, and your thoughts feel softer, slower, like feathers settling on still water. And somewhere inside you, beneath layers of breath and blood, the spiral hums on. It carries your story. But tonight, the story begins elsewhere, not in you, but in something much smaller, much older, much wiser in its own simple way. Let us drift there now. Float backward in time, billions of years, to a world of heat and salt and storm. There, among ancient oceans, life stirs in its earliest form. Bacteria, tiny cells, no nucleus, no brain, but alive, alive, and clever. They live in numbers beyond imagining, suspended in water, clinging to rocks, floating on invisible currents. And yet, even in this quiet, they are under threat from viruses, not the kind that bring fevers to your body, but the kind that haunt the single-celled. They're called bacteriophages, phages for short. They are sharp, geometric things, like alien probes, all legs and tails and hunger. They do not knock. They stab. They inject their code, forcing the bacterium to become a factory for making more phages. And in moments, the cell is filled, bursting apart in a viral bloom. But bacteria are not helpless. In the arms race of life, even the smallest find ways to fight. And here is where something extraordinary happened. Bacteria evolved a memory, a kind of molecular notebook where they stored the genetic mugshots of their enemies. A defense system written not in weapons, but in awareness. Imagine a scroll inside the bacterium, a stretch of DNA where it pastes in fragments of the viruses that attacked it. Just tiny snippets, just enough to remember. This scroll became known as CRISPR, clusters of repeated code with spacers in between, each spacer a stolen piece of a viral attacker. The bacterium keeps this list like a record of past battles. And when danger returns, when a matching virus appears again, the bacterium consults its memory. It transcribes the matching sequence into a strand of RNA, a tiny guide, and it pairs that guide with an enzyme called CAs9, its guardian. CAs9 is the sword, but it swings only when told where. It needs the guide to see. Together, they drift through the cell, searching every corner, every molecule of DNA. And when they find a match, when the invader's code matches the memory, CAs9 slices it apart. Clean cut. The virus is stopped. The cell survives. This, then, is the original CRISPR. Not a human invention, but a bacterial wisdom, a living immune system built on memory. And here's the wonder. This defense is heritable. When the bacterium divides, it passes the scroll on. Its children remember what it fought. They are born with knowledge, earned by survival. Can you feel how elegant that is? A cell with no eyes, no ears, no mouth, and yet it remembers. It learns. And in that silent remembering, the foundation of modern gene editing was born. Scientists didn't invent CRISPR, they discovered it. They looked into the bacterial world and saw something astonishing, a system not only to recognize DNA, but to cut it with precision. And so they borrowed it. They took the same guide RNA, but instead of matching a virus, they made it match a human gene. And instead of destroying an invader, they used Cas9 to correct mutations inside our own cells. This is the heart of it. Nature already solved the hardest part: the recognition, the specificity, the exactness. We only needed to redirect the tool. This tiny bacterial guardian, once tasked with defending a cell from death, now helps doctors explore cures once thought impossible. It's poetic, isn't it? That a single-celled organism, living long before we walked the earth, held the key to editing the code of life. That something so ancient could become something so modern. And yet, even now, bacteria still use CRISPR for the same reason they always did: to survive. Their guardians still patrol the spiral, still carry their guides, still remember. CRISPR has not changed. It has simply expanded its role. It serves us now, too. But there's a humility in remembering where it came from, because for all our brilliance, we are not the origin. We are observers, borrowers. We stood at the edge of a tide pool and saw a defense mechanism more elegant than anything we could build ourselves. And we listened. We translated it into medicine, into therapy, into possibility. But we must also carry its lessons. The bacterial guardian was not designed for creation. It was designed for caution. It cuts only when the match is perfect. It waits. It listens. In that same way, we too must move with care. CRISPR is sharp. It is fast. But it is not wise on its own. It needs intention. It needs purpose. And as we begin to use it, not only to heal, but to design, we must carry the memory of where it came from. A quiet cell, drifting in the ocean, facing a deadly virus, and choosing to remember. That was the beginning. And now, we must ask ourselves, what will we choose to remember as we carry this tool forward? What lessons from the smallest life should guide our choices in the largest? In the chapters ahead, we'll begin to face those questions, because the power to edit genes is no longer theoretical. It's here, real, already being used. And the next step is not scientific, it is human. You are resting now, still, but listening. Your breath moves slow and smooth, like a river untroubled by wind. In this quiet, you are ready to step softly into a tender place where science and care must walk together. Because tonight's story is not about what we can do, but how gently we do it. You already know the spiral, the curled staircase of your DNA, each step a chemical letter, each turn a part of you. And you've met CRISPR, the bacterial guardian reborn, now guiding a scalpel of precision through the library of life. But now, the question deepens. How do we use this tool without harming the page it edits. You see, genes are not simple switches. They are more like musical notes, playing not in isolation, but in chords, in harmony, in time and rhythm with others. A single change, a single snip, can echo in ways we do not expect. And so, to edit life is to enter the music with humility. Imagine a sentence written in light. She will walk beneath the trees. Now imagine changing one word. She will run beneath the trees. She will fall beneath the trees. She will fly beneath the trees. Each version is still a story, but each changes everything. This is what gene editing feels like. The shift of a single base pair, a single letter, can turn a life. And that's why we do it gently. In labs around the world, scientists wear gloves not just for sterility, but for reverence. They look into microscopes and see not just cells, but futures, children not yet born, diseases not yet awakened, hope still folded in silence. And when they guide CRISPR into a cell, they hold their breath because precision matters. The enzyme CAS9 makes a cut. It's guided by a strand of RNA, a map to a very specific location. The cell's own machinery responds, rushing in to patch the break. And here's where we can guide it. Adding a corrected piece of DNA, a patch of healthy text, to replace what was flawed. It's not always perfect, but when it works, it's like watching a lock click open, something long stuck, suddenly released. This has already changed lives. Children born with sickle cell disease have had their bone marrow cells edited and their symptoms eased. People living with certain forms of blindness have had their retinal cells gently corrected, bringing light back into their world. And trials are underway to edit immune cells, to target tumors, to quiet the genes that allow diseases to grow unchecked. This is not science fiction. This is medicine. Today. Now. But we must be gentle, because even in healing, we are changing something fundamental. The cell did not ask to be edited. It trusted its code. And when we intervene, we are rewriting an old letter with hands that must be steady and hearts that must be wise. You might wonder, what happens when we edit too much? What if we don't stop at healing? What if we begin to design? Height, by color, memory, mood. Where is the line between medicine and modification? No one can answer this easily, but many are trying. Bioethicists, doctors, lawmakers, sitting at tables, arguing gently, raising questions like candles in a circle. They are asking, who gets access to this technology? Who decides what's a defect and what's a difference? What are the risks of editing not just the individual, but their descendants? Because when you edit the germline, the sperm, the egg, the embryo, the change becomes permanent. It is passed down. It becomes a thread in the human tapestry, and that thread cannot be pulled back. That is the weight of this moment. CRISPR is not just a tool of repair. It is a tool of possibility, and possibility is delicate. It must be carried gently, like a seed that may grow into anything. We are standing now at the very beginning of a long path. Each footstep must be made with care. The edits we make today will echo not just in one life, but in the children and grandchildren of that life. And so we must ask ourselves, are we correcting suffering or are we chasing perfection? And what does perfection mean? Whose standard do we use? These are not scientific questions. They are human ones. To edit a gene is not to fix a machine. It is to enter the story of a person. And people are full of unknowns. Their beauty is not always in symmetry. Their strength is not always in standard form. Some of the most extraordinary lives have begun with mutations, with differences, with struggles that became songs. So as you drift deeper into this quiet night, hold on to this thought. To gently edit life is to approach it like a gardener, not a sculptor. A gardener listens. They don't force the rose to become a tree. They watch, they tend, they care for what is, while nurturing what could be. CRISPR, when used with love, can be that gardener's hand. But it can also become something harsher if guided by fear or pride or the illusion that we can perfect what was never broken. Let the spiral speak. It has wisdom older than us. And as you drift now, still wrapped in warmth, still resting in wonder, we begin to feel the weight of the next question rising. If we can change a child's future before they're even born, then how far will we go? And who gets to decide what kind of future is worth choosing? Sleep lies just behind your eyes now. You're still and warm. Your breath so steady it feels like a tide, slow and certain, moving through your chest like waves through a moonlit bay. Let yourself drift now, not into the past, but toward the edge of possibility. Because the chapter tonight is not one we read, it's one we might write. We are standing at the threshold of a new kind of dreaming. Not the kind that happens in sleep, but the kind that takes place in white-walled labs beneath microscopes, where hands steady over petri dishes and futures are shaped cell by cell. Designer babies. The phrase arrives like a spark, bright with promise, but casting a long shadow. To some, it sounds like salvation, an end to inherited illness, a child born free from suffering before their first breath. To others, it whispers of something colder, a child crafted to spec, chosen, not simply born. But tonight, we are not rushing to answer. We are here to feel the quiet weight of the question. You already know CRISPR. It can seek and edit a gene with astonishing precision. And when used on embryos, before the first heartbeat, before the brain folds, before the body even begins, those edits ripple outward through every cell for the rest of that life and through every life that follows. This is called germline editing, and it changes everything. Because this isn't just healing the sick, it's shaping the not yet born, writing before the first word, drawing the map before the traveler walks it. It begins with good intentions. What parent wouldn't want to remove a terrible disease from their child's path? To take away the gene for Huntington's, for cystic fibrosis, for a cruel, silent blindness? To take the spiral in their hands and smooth it gently. But once we begin, where do we stop? If we can remove disease, can we also add resilience, boost immunity, sharpen memory, lift mood? And then, can we choose height, eye color, temperament? Can we tilt the odds towards certain talents, certain appearances, certain lives? And who gets to decide? This is not speculation. The science is moving forward. In 2018, a scientist in China used CRISPR to edit embryos. Twin girls were born, with changes meant to grant them resistance to HIV. The world was stunned, not just because it was possible, but because it had been done without consensus, without safety, without clear ethics. It felt like a door had opened in the dark. And yet, it is not evil to want health. It is not wrong to dream of a world where no child suffers from inherited pain. But we must be careful not to confuse health with preference, not to trade complexity for control. Nature does not deal in guarantees. Even with perfect genes, life is still unpredictable. A person is more than their DNA. They are shaped by time, by love, by chance. To dream of designing a child is to imagine we understand more than we do. Genes do not work in isolation. They interact like dancers in a crowded room, stepping around each other, bumping, influencing, responding. Change one, and the others may shift, sometimes subtly, sometimes with consequences we could not have seen. And then there is the deeper question: should all parents have this choice? Or only the wealthy? Does this technology widen the gap between privilege and poverty? What happens when the designed child grows up in a world where others were not? What burden does that child carry? To be optimized, to meet expectations they never chose? What if they don't want the future their genes were written for? There is a gentleness in not knowing, a grace in the randomness of birth. Each child, a mystery unfolding. When we design, we may narrow that mystery. We may lose the wild, the unexpected, the wonder. Still, we are here now with this power in our hands, and we must decide what it's for. Do we use it to lift pain from those born into it? Or do we begin to craft lives in our own imagined image? Do we act as stewards or architects? And how do we protect the future from the unintended consequences of our very best intentions? You don't need to solve these questions tonight. You're here only to rest in the softness of the asking, to let your breath be slow and your mind gentle as you imagine the embryos, so small, so silent, holding potential beyond measure. They are not blank slates. They are tiny symphonies, not yet played, and we must decide whether to compose them or simply listen. The science is here. The dreams have begun. And now, so must the dialogue. Tomorrow will bring debates, panels, protests, promises. It will bring laws. It will bring mistakes. It will bring beauty, too. But for now, you rest. You let the questions float like petals on a stream, not needing to grasp them, only to feel their passing shape. Because in the next chapter, we step into the living room of the future, where families must choose. Not in theory, not in research, but in real life. We meet those who stand at the edge of these decisions: parents, doctors, counselors, trying to weigh love against uncertainty. And so we drift forward now toward the moment of choice. You are floating now, softly between sleep and waking, where thought becomes mist, and time feels wide and slow. Your breath is gentle, your body is safe, and your mind, your beautiful, wandering mind, is drifting toward your quiet room, not in a lab, not in a simulation, but in a hospital. There is light here, warm, fluorescent, pulsing in still air. There are people, nurses, moving softly, monitors glowing. And in the center of this room, a cry, a newborn cry, sharp, small, real. It is not the sound of science. It is the sound of a life beginning. But this child, this small girl, now learning to breathe, is unlike any child born before her, because deep within her, CRISPR has already been, not to heal, not after birth, but before she was even an embryo, in the earliest hours of her becoming. A scalpel made of light rewrote her code. It was done by a scientist named Tijiankui in China, in secrecy. He edited a gene called CCR5, one that helps HIV enter cells. He hoped to make her resistant to the virus, just as some people naturally are. He called it protection, but many others called it something else. Reckless, premature, a line crossed without consensus. When the world found out, it trembled, because this was no longer an idea. It was a person. And once a person exists, there is no going back. She was given a name, Lulu, and soon after, her twin, Nana. Two babies, born into controversy, born into a storm. They had no part in choosing. And yet, in that moment, in that first cry, none of that mattered. Only the fact that they were alive, that they were whole, that they were breathing. Still, scientists everywhere stopped in their tracks. Some wept, some raged. Many asked, how could something so powerful be used so carelessly? Because the edits had been done with imperfect knowledge. The girls weren't at risk of HIV. Their parents were carriers, but they were not infected. There were safer ways to prevent transmission, medical ways, ethical ways, and the gene-edited CCR5 was not simple. It affects the brain, the immune system, perhaps even memory and learning. The science wasn't settled, not yet, but the edits had been made. The scissors had been guided, the DNA cut and patched. And now, two children would carry that change for the rest of their lives. Not only them, but their children and their children's children. This was not a treatment. It was a transformation. And the world wasn't ready. Regulations tightened. Conferences filled with debate. The scientist was condemned. His work halted. His reputation shattered. But Lulu and Nana remained. Two lives. Not ideas. Not warnings. But daughters. We don't know where they are now. Their identities were hidden to protect them from the public eye. And perhaps that's for the best. Because what they need is not scrutiny, but safety. Still, we remember their birth as the moment when the dream became reality. It changed the question from, should we, to, we already have. Now what? Because the technology cannot be unmade. The ability to edit embryos now exists. What remains is the responsibility. The first CRISPR baby cried, not in triumph, not in tragedy, but simply as any baby cries, hungry, confused, alive. But her cry echoed into halls of power, into scientific journals, into bioethics boards, and into the dreams of future parents who now wondered if their child, too, could be shaped. And so now, the conversation has changed. We are no longer imagining possibilities. We are managing consequences. And we must ask, what does it mean to be the first? Is she a miracle? A warning? Both? And what of her humanity? Does it change because her DNA was altered by human hands? Of course not. She is still stardust, still breath and laughter and dreams. But her existence reminds us that power and wisdom do not always arrive together. To hear her story is not to condemn CRISPR. It is to remember that every tool reflects the hand that holds it. In the hands of care, CRISPR could cure, could heal, could set lives free. In the hands of haste, it could wound unseen, could alter what should not be touched. Even the name CCR5 is a reminder that genes are not just letters. They are doors, open, closed, or reshaped. And when we edit them, we are stepping through. So now, as you rest deeper, ask not just what CRISPR can do, but what it should be for. Ask how we protect those who are edited before they even speak. Ask how we honor the children whose cells were changed before their first smile. Ask how we keep wonder without losing caution. And most of all, ask how we listen, not just to scientists, but to parents, to ethicists, to children not yet born. Because even now, in their silence, Lulu and Nana speak volumes. Their birth is no longer an experiment. It is a story unfolding, and we are all turning the pages. They will grow, they will dream, and the world will watch, quietly, curiously, wondering what it has begun. And as you slip toward sleep, held in the hush between dreams, let the sound of that first cry echo softly in your mind. Because in the next chapter, we step into the hearts of parents who now carry this power in their hands and must choose whether or not to reach for it. You lie still now, wrapped in quiet, a silence thick and slow, like morning fog. The cry from the CRISPR nursery still lingers in your mind, but tonight you follow its echo into chambers where decisions are whispered before they are made. This is not a place of noise, not a place of clashing metals or spinning centrifuges. This is a place of thought, a space where soft minds meet beneath dim lamps of uncertainty. Yeah. Answers don't shout. They arrive as questions, slow and trembling, like rain beginning on glass. Welcome to the ethics of gene editing. You find yourself seated in a circle that isn't quite visible, but deeply felt. It holds scientists, philosophers, mothers, fathers, children grown, and elders whose faces carry decades of wondering. They speak slowly, their voices woven into the fabric of something fragile, consent. One voice, low, steady, asks, what does it mean to choose for someone who cannot speak? Another follows, if we can prevent suffering, is it not our duty? Or does that very act risk becoming its own form of harm? And then, when does healing become enhancement? When do we stop saving and begin designing? You breathe deeply now. This is not debate. This is listening. This is holding silence without rushing to fill it. In the center of the room, not made of matter, but very real, spins the double helix, graceful, unfolding. Still, it does not beg to be changed, but nor does it resist. It simply turns, as it always has. And so the question remains, not in the molecule, but in the mirror, in us. You imagine a parent gazing into a microscope, seeing a cluster of cells, fragile, luminous, their future child. The doctor offers a path, a single change, a fix. No more cystic fibrosis, no inherited blindness, no early death. Their heart aches with love. They want to choose life, a better one, a freer one. But what does better mean? And who gets to define it? Ethics whispers gently. What happens when suffering is redefined by society instead of by the one who lives it? You see a mother who chose not to edit. Her child, born with a rare condition, laughs like birdsong. She speaks of time as something slowed, something deeper than she knew before. You see another parent who did choose the edit, but the outcome was not what they expected. The trait was removed, but something else emerged. A silence in the child's eyes no one could explain. A soft voice in the circle, a bioethicist, says, CRISPR is not magic. It is precision, but not perfection. The future we design is not always the one we receive. Now you see the pillars, etched not in marble, but in principle, four truths held close by those who guide us: autonomy, beneficence, non-maleficence, justice, autonomy, the right to choose. But whose choice matters when the chosen one has no voice? Beneficence, to do good. But good is shaped by culture, by fear, by longing. Non-maleficence, do no harm, yet harm often hides until it's too late. Justice, to be fair, but fairness requires a world that treats all equally, and ours does not. And still, the helix spins. You imagine a future rushing forward, children chosen for brilliance, for beauty, for traits selected like groceries in a cart. What becomes of the unexpected child? The child not designed, but simply born? Will they be celebrated or pitied? Will they be seen as whole or as a missed opportunity? And yet, you also see mercy. You see a parent who ends a family's line of illness with one choice, one snip of molecular scissors. You feel no judgment, only weight, only tenderness for the human heart trying to protect what it loves. You drift past headlines, debates, laws being written in high towers, but return to this soft circle, this place where voices do not rise in anger, but kneel in curiosity. You hear one last voice, older, wiser. She speaks gently. We must not edit away mystery. Let there still be room for surprise. And with that, the chairs fade. The voices hush. The circle dissolves. The double helix remains, spinning slowly like a mobile above the crib of our shared tomorrow. And you, still cradled in thought, float onward to the next question where love and choice entwine. Who, in the end, gets to decide? You float quietly now, beneath the soft breath of time, where dust gathers on paper and memory settles like ash. This is a different kind of laboratory. No microscopes here, no gleaming white walls, only old words in typewriter ink and silence that carries a deep and painful echo. You walk through history tonight, and the past watches. It does not scold. It does not shout. It simply waits for you to remember. You come to a door with a name carved faintly. Eugenics. The word is dry in your mouth, but heavy. You open it anyway. Inside, there are charts, files, measurements of skulls, forms signed by trembling hands. Here, science was used not to heal, but to separate, to rank, to erase. 100 years ago, people in coats spoke of purity, of betterment, of cleaning the gene pool, like one would cleanse a river. They used science to decide who should be born and who should not. And many, so many, never had the chance to live because someone else thought they weren't good enough. You feel it now, a shiver beneath the skin, a recognition in your cells, because what began as a whisper of improvement became a scream of control. It began with terms like feeble-minded, unfit, undesirable. Then came forced sterilizations. Then came silence. Then came names, lives lost to decisions made in clinics and courtrooms. And the worst part? It was called science. It wore a lab coat and carried a clipboard, the same tools we now use to heal once harmed. You walk through archives where hope was twisted into hierarchy. A letter from a mother pleading not to have her daughter taken. A chart that ranked people by value. A law that allowed it. This is not ancient history. This was yesterday, in the lives of some still living today. And now you return to the present, CRISPR in hand, bright as lightning, in a midnight room. You ask, what protects us from walking this path again? The answer isn't found in the science itself. Science has no conscience. It follows the hands that guide it. The answer is in remembrance, in the names we refuse to forget, in the harm we promise not to repeat. You sit in that quiet now, not with guilt, but with awareness, because awareness is armor, and ethics is memory, emotion. You think of a child who might be born with different abilities, a mind that moves sideways, a body that doesn't match the norm. Is that a problem to be solved, or a person to be met? In the wrong hands, editing becomes erasure, a quiet violence wrapped in the language of progress. So you hold the question gently. What kind of world are we editing toward? World without suffering, or a world without difference? And are we wise enough to know the difference? You remember now, in early days of genetics, The word fitness was borrowed from evolution and used to justify cruelty. But evolution is not about perfection. It's about diversity, adaptation, resilience born from the unexpected. The orchid and the cactus both bloom, but only one thrives in the desert. Sameness is not strength. You drift now through stories of resistance. A midwife who hid records to save a family. A doctor who refused to comply. A grandmother who whispered truths when the world told her to stay silent. Their courage was not written in textbooks, but it lives in the hands of those who do science with care. You picture a classroom where children learn about genes, not just as codes, but as legacies of survival and struggle. You picture a conference table where disabled voices are heard first, not last. You picture a parent who says, I do not want to erase my child's difference. I want to build a world where they can thrive in it. The ghosts of eugenics do not haunt you to scare you. They walk beside you to remind. They say, if you are to carry this torch, carry it with humility, with curiosity, with kindness. Let love be the compass. Let the child be seen before the trait. Let the question always come before the cut. And let science never forget its human face. You float upward now, leaving the archives behind, but not forgotten. They remain etched into the ethical spine of tomorrow's science. And above you, the double helix turns, not pure, not perfect, but alive, twisting in all directions, holding stories of pain and beauty in the same breath. You lie still, but your thoughts wander gently toward a place where science moves, not alone, but arm in arm with history. Because in the next chapter, a new question waits, not one of harm, but of hope. Can science repair what was once broken? Can healing reach backward as well as forward? You exhale slowly. The ghosts fade. The future remains. Listening. You are lying still now as the hush of night pulls soft shadows across your thoughts. Above you, the stars stretch wide, but tonight you look inward. You drift into a garden, not of flowers, but of life itself, the gene pool. Not a pool of water, but a river of possibility flowing through generations, from the deep past into the unborn future. Each life, each seed, a unique ripple in that river. And here, in this garden, the air is warm with memory. You walk barefoot along soft soil, past winding rows of invisible roots. This is the place where variation grows, where difference is not a flaw, but a design. The gene pool is vast, ancient, and filled with echoes. Some genes carry songs of survival, resistance to disease, endurance in cold, wisdom in strange forms. Some carry risk, yes, fragilities handed down, like heirlooms. But even these have their stories. You see a flower that grows with curled petals. It leans to the side, but in its curve, it collects rain more gently than any other bloom. Another flower, pale and awed, feeds a bee that no other plant will feed. Diversity, you realize, isn't just beautiful, it is functional. Resilience lives in variety. You walk deeper. You see a gardener now, someone like you, with hands covered in earth, heart full of care. She kneels beside a seedling with twisted leaves. She does not pull it. She studies it. She wonders why it twists. She waits. This gardener has learned that not every crooked stalk needs straightening, and some do. A stalk that cannot stand may long for light it cannot reach. And so she helps it gently with a slender stake and loose ribbon. She does not change the seed. She listens to it. This is what tending means. You sit beside her, watching. She tells you softly, the garden grows wild for a reason. Too much order, and you lose the bees. Too much sameness, and the soil forgets how to breathe. You remember CRISPR now, that small, sharp scalpel of light, and you wonder, what kind of gardener do we wish to be? Do we pull every weed? Or do we learn which ones are wild herbs? Do we aim for roses alone? Or do we leave room for dandelions and moss? The gene pool, this river of inherited possibility, is not something to clean, but to protect. There are alleles that once seemed broken, now known to offer unexpected strength. A mutation that resists malaria, a gene that protects against HIV, an adaptation for high altitudes in the lungs of those born on mountaintops. What we once called defects may be dormant gifts. You walk now through a section of the garden where stories grow. A child born with a rare syndrome who paints galaxies from memory. A boy with brittle bones who becomes a poet of touch. A girl who stutters but sings with flawless clarity. Each one a variation, not broken, just rare, just different. You pause now. The path splits. One side leads to symmetry, rows of perfect crops, identical and gleaming. The other leads into a wild grove, where vines twist and petals spill with no plan but life. You ask yourself, which one feels more alive and which one feels more fragile? Uniformity looks strong, but it breaks easily, like a field of clones that fall to one storm. But diversity endures. It adapts, it bends, and begins again. The gardener smiles. She tells you, we can use tools like CRISPR to help the struggling plants, but not to erase the garden soul. You think now of the term gene pool. It sounds so clean, but it is messy and beautiful and crowded with life. A pool that reflects our past and future both. Every step you take in this garden is a step between what is and what could be. And so the question grows, are we caretakers or engineers? Can we make space for imperfection and still offer healing? You kneel in the soil. Feel the earth's warmth beneath your fingertips. This is where all of us began. From scattered seeds, from mutations that stuck, from errors that turned into innovation. Every feather, every fin, every face, sculpted by time, by pressure, by chance. And now we hold the tools to sculpt ourselves. The gene pool is not a blueprint. It's a living library, a garden, yes, but one written in stars and stumbles. What we prune, what we protect, what we choose to edit, these choices echo for lifetimes. And so you slow your breath, you listen, because the gene pool is not asking for perfection, it is asking for permission to be alive. You rise now, heart calm, the night air sweet with unseen pollen, and as you leave the garden, you carry a seed in your palm, not to change, but to understand. For the next chapter waits at the edge of the field, where hope begins to wonder aloud. Can we use CRISPR not only to edit genes, but to restore what was once lost? Not just to cure, but to remember? And in that soft wondering, your thoughts gently drift onward into restoration, into repair, into the soft green hush of what might bloom next. You are quiet now, nestled in stillness, your breath slow, your thoughts open like moonflowers in the dark. Tonight, you walk softly through the unseen, not the galaxies far above, but the galaxies within, the swirling codes that shape your blood, your bones, your being. This chapter is not about fixing what is broken on the outside. It's about healing what hides. You drift into the quiet hallways of the body, into a place too small for sight, but big enough to carry pain through generations. Inside your cells, there are words misspelt by chance, fragments misfolded in the long poetry of your DNA. Some of these errors are harmless, but others, they whisper in blood. They steal breath. They twist the body into quiet battles no one can see. You arrive now at a room of children, not sleeping, waiting. Some wear tubes, some wear smiles. All of them carry something inherited they never asked for. Sickle cell, Tay-Sachs, muscular dystrophy, cystic fibrosis. Their illnesses don't show in photographs, but they live in every breath, every step, every moment. Missed. These are not rare names in medical books. They are lullabies interrupted, school days spent in hospitals, dreams paused by genetic weight. You sit beside one child. Her name is Maya. She's 8, and her blood cells bend into crescent moons that block her veins. Pain comes without warning, and yet she smiles when she talks about space, about stars, about becoming an astronaut. Her mother holds her hand, quiet and tired. And in this moment, you understand what CRISPR offers, not a miracle, but a tool, a chance to reach into the place no medicine could reach before, to find the misspelling, to rewrite the letter, to restore the sentence, not for perfection, for peace. You move now into the laboratory, a room that glows with purpose. A tube holds a drop of Maya's blood. Inside, her stem cells sleep lightly, waiting. A CRISPR molecule drifts close, guided like a compass, toward one broken phrase. It does not scream. It does not burn. It cuts gently. It offers a new letter. The edit is small, The result, immeasurable. If it works, or blood may flow like a river again. No more hospital beds. No more waiting. Just life. You feel the weight of this. Because healing here is not only about one child, but many. You picture families who've watched generations carry the same pain, a story written in the same flawed line of code. Now, CRISPR whispers, maybe the story can change, but healing the unseen is not simple. There are risks. Some edits cause new ripples. Some cuts leave scars. Sometimes the scissors snip the wrong strand. And so, scientists move slowly, like careful gardeners, pruning leaf by leaf. You meet a researcher. Her name is Dr. Yara. She has spent 10 years on one disorder, one gene. She tells you this work is slow because it must be sacred. You watch her hold Maya's sample as if it were a lullaby in glass. Because when you heal at the level of the gene, you are not just treating illness, you are rewriting inheritance. And what we inherit defines not only our bodies, but our futures. You drift now into a deeper place, into the brain. There are disorders here, so delicate, they slip through language. Autism, epilepsy, schizophrenia, conditions with roots in many genes, many moments. Not one cause, but a mosaic. Here, CRISPR is more whisper than cure. Still, there is hope that one day, even these may become gentler, if not erased. But the line is fine, because what is difference and what is disorder? When do we heal and when do we silence what was never meant to be gone? A poet with synesthesia sees music in colors. A child with Tourette's becomes a composer of rhythm. A man with bipolar disorder writes stories that change lives. If you edit the gene, do you edit the gift? CRISPR does not answer. It only offers. And you hold a choice. You float back now to Maya. Her trial is ongoing. The edit was made. And for the first time in memory, she laughs without pain. She runs, not just in her mind, but across her backyard, beneath stars. She still plans to reach. Her mother weeps into the wind. You see now, healing the unseen is not about magic. It is about precision, compassion, and time. CRISPR is not a cure-all. It is not always the answer. But sometimes, for someone, it is the key to a locked door. A door they never thought they'd walk through. You lie still now, your body breathing on its own schedule, your thoughts gentle. Somewhere inside you, your own cells carry code. Some letters clear, some silent errors you will never know. And still, you are here, alive, unfolding. You smile, because the promise of healing is not just in the cure, but in the care. And in the next chapter, you will follow that care into even stranger places where CRISPR reaches, not into the body, but into memory, into lineage, into long buried pain. And you wonder now, can we heal the past itself? You drift quietly, your breath long, your spirit warm. carried onward into the arms of inheritance. The night is deep now. Your limbs are still. Your breath flows like a tide, gentle, returning. And here, in this stillness, a fork appears. Two paths, two doors, two futures. You do not choose yet. You simply look. One path glows softly. It leads to the present body. This is somatic editing. The other path is older, winding deeper into the soil of time. This is germline. You begin with the first. You walk the path of somatic cells, the cells of your skin, your blood, your lungs, your bones. These are the workers, the doers. They carry your life forward, each hour, each breath. When a gene is edited here, the change stays with you, only you. It does not travel forward to your children's children. Somatic edits are surgical, personal, temporary in the river of lineage, like trimming a single branch on a vast, old tree. You reach a clinic now where a patient waits. He has a muscle disease. His body weakens, though his mind remains a lantern in the dark. A single gene, misspelled in the body, but not in the spirit. Doctors use CRISPR here to fix the spelling in his muscle cells. Not forever, but enough to hold him upright. Enough to dance at his sister's wedding. Somatic editing offers relief without rewriting the future. And yet, you feel its limits. It cannot stop the gene from returning in the next generation. It treats the leaves, not the roots. And so, you walk back now toward the second path. This one is quieter, older, heavier. This is germline. Here, the edits are made not to the body you walk in, but to the seeds of life, to sperm, to egg, to embryo, to the pages of inheritance before they've been read. You walk into a lab now. A dish holds an embryo, barely hours old. Its cells divide like quiet firelight. And in those early flickers, a scientist introduces CRISPR not to heal this person, but to change all who follow. Germline editing reaches into the infinite hallway of tomorrow. One change echoes down the line. One edit becomes legacy. This is power rarely held, and it asks for hands gentler than ambition. You pause here, because this is no longer about treating disease. This is about shaping who gets to be born and how. A single letter changed here may erase a disorder or erase a difference. You see a mother now. She holds a choice. Edit the embryo to remove a gene that carries deafness. Doctors say it's possible, safe, but she hesitates because her family signs, because music to them is the movement of hands, because silence to them is not absence, it is language. What is a disorder to one may be identity to another. You step softly now through this space between healing and altering, because germline edits are permanent. They ripple into great-grandchildren never met. They promise freedom from suffering, but also ask, at what cost to the wildness of human variation? You return now to the crossroad. Somatic to the left, germline to the right. One path treats, one path transforms, and somewhere between them, you feel the soul of CRISPR, a tool with no will of its own, a light that can illuminate or blind. You think now of ancient roads, how every civilization built forks like this to build the dam or let the river run, to cross the sea or stay rooted. And now our fork is microscopic. A single gene, edited in the dark, can guide millions of lives. And yet, we are not only bodies, we are culture, we are error, we are anomaly and adaptation folded into each other. What if some of our greatest gifts come not from the edited, but from the unexpected? You sit now between the paths. No need to choose, only to wonder. Because the question is not only, can we, when, for whom, and why. Somatic edits give us tools for comfort and care. Germline edits offer visions of redesigned futures. But the world is not a draft, and we are not blueprints. You breathe in and out, because the point of science is not always to move fast, but to move wisely. And the point of CRISPR is not to perfect humanity, but perhaps to protect it. In both paths, there is potential. In both, there is risk. But in your quiet pause, there is something deeper. The recognition that just because we hold the pen doesn't mean we understand the story. And as the moonlight pours onto the crossroads, you walk forward slowly toward the next chapter where lines blur between nature and nurture, where the question is not what we inherit, but how we live with it. And your breath, gentle now, carries you there, into the soft soil of identity. You're cradled now in the hush between thoughts, where time folds softly like a blanket around your shoulders. And as you drift, you arrive at a place gentler than a lab, a nursery. But here, the cradle holds not just a child, but a question. It is this: can someone consent before they are born? You do not answer quickly, because this question is not sharp, it is slow, like the quiet swing of a mobile above a crib. You sit down in the room of beginnings, where embryos rest, like unopened letters. Each one a possibility, a path not yet walked, self not yet aware. And beside them, a scientist, a parent. I hope they want to help, to correct, to prevent, to choose. For someone who cannot speak, the intent is love. But love, too, must ask. You listen now to the voices of ethics, returning from the circle you met chapters ago. One says, it is not wrong to want to help, but we must be sure we are not helping ourselves more than the child. Another asks, can there be consent without consciousness? You feel this deeply, because you, too, were once a whisper, cells dividing with no name, and everything you are now was once chosen by someone else. What music you heard, what words you learned, what world you entered, none of it was your doing. But your genes, your very structure, that, until now, was untouched. CRISPR changes that. It moves us from parenting to pre-planning, from nurturing the child to shaping the child before they take their first breath. And while this offers rescue from suffering, it also removes the child's ability to one day say, I would have chosen differently. You meet a young woman now. Her name is Isla. She is 17, bright, kind. She was edited as an embryo. Her parents chose to remove a gene linked to mental illness. She is healthy, grateful, even. But she says softly, I wonder who I would have been if they had left that part of me in. She does not resent them, but she carries a strange inheritance. Not a gene, but a decision she could never weigh in on. You walk with her, through her memory, through the clinic brochure her parents read, through the consultation, the hope, the signatures. Nowhere, nowhere was her voice, because it could not yet exist. You see the complexity now. Consent is the cornerstone of ethics. We ask before touching, before sharing, before treating. But with germline editing, we act before the subject exists. And so we must become caretakers of future will. We must imagine the voice of someone not yet formed. Would they want this? Would they thank us? Or would they say, you edited away what I might have used to understand myself? You pause here, because not all edits are equal. Some remove known suffering, a gene for a fatal disorder with no cure. Others, they tread closer to preference, to enhancement, to comfort, for the parent more than the child. And so consent becomes blurry. It becomes a mirror, reflecting both care and control. You hear now a story told in hush. The couple chose to edit for taller height. Their child, now 12, wonders aloud, was I not enough the way I might have been? You breathe in, because this is not a question for laws alone. It is a question for love. And love, true love, listens, even when the voice is silent. You step now into a circle of doctors, ethicists, parents-to-be. They are building a framework, not just of rules, but of respect. They ask, should edits be limited only to disease? Should every child have a right to an unedited self? Should there be a pause, counsel, a ritual of reflection before any cut is made? And how do we include the child's future voice in a decision made long before they open their eyes? There are no easy answers, only the gentle truth that science must always be guided by listening. Even when the listener is not yet born, you return now to the cradle. It is quiet, peaceful, unaware, but you know within it is a story that will unfold, a life that will ask questions one day. And we must be able to answer honestly. We did this because we believed it was right. We paused. We questioned. We cared. Not just about your health, but about your wholeness. You step away now. The night wraps you gently. Because this chapter was not about answers. It was about pausing to hear the silence, to honor the unborn voice. And as you drift now into the next dream, you follow that voice, soft and rising, into a new chapter, one that asks, what happens when genes are no longer private? What happens when the story of your DNA is sold? And so you breathe, slow and wide, and step gently into the shadowed space of data, privacy, and power. The wind is different here, cooler, wider, filled with the scent of soil and pine. You've left the clinics, the microscopes, the silent nurseries. Tonight, you walk under stars, beside rivers, through meadows lit only by fireflies and breath. This is where CRISPR has wandered, not just into bodies, but into the wild. You take soft steps through grasses that hum, and above you, a moth circles slowly, its wings painted by a gene not found in any natural history book. You are entering a new kind of nature, a place where DNA has been edited not in the name of healing, but in the hope of repair. Because the wild, too, is wounded. Species vanish. Pests overwhelm. Diseases leap from animal to orchard to village. And here, in this tapestry unraveling, CRISPR has been offered as a needle, a way to stitch back what's come undone. You kneel by a field of wheat, its stalks tall, golden, strong, but within its cells is a change. It resists blight through an edit too precise for breeding, too swift for tradition. A single snip, and the harvest is saved. A farmer nearby watches the sky. She says, the rains don't come like they used to. The soil gives less each year. CRISPR didn't make my field perfect, but it gave us time. You rise, grateful and wary, because time is a gift, but not always a solution. You walk on into forests darker, older. Here, an invasive beetle has hollowed the hearts of 1,000 ash trees. Scientists released a countermeasure, beetles edited to fail at reproduction, a generation designed to end itself. And for now, the trees breathe again. But you ask, what else changes when the beetles go silent? The fox that feeds on them? The bird that nests nearby? The fungus waiting to fill their absence? In nature, no change stands alone. A single gene is a thread in a web. Pull it, and you must watch what trembles. You come now to a lake, still, dark, reflective, and in it, mosquitoes, swarming, but not for long. Scientists have tested what they call a gene drive, a way to force a gene through a population so quickly it overtakes evolution. CRISPR makes it possible. One release, one mosquito, and soon a region cleared of disease. No malaria, no dengue, no Zika, but you feel a chill because the same tool that saves can also silence. And once set in motion, a gene drive cannot be recalled. The wild does not sign waivers. It does not hold meetings. It cannot consent. And yet, it receives our edits quietly, completely. You sit now beneath an acacia tree. A researcher joins you, her boots dusty, her voice tired. She says, we edit to heal ecosystems, to restore balance. You nod, but she pauses, and sometimes she adds, "We do it just because we can." You feel that truth. CRISPR's beauty is its simplicity, its ease, once reserved for scientists in glass towers, now in kits on kitchen tables. Edit a mushroom to glow, edit a pet to shimmer. What begins with restoration may end in recreation. You close your eyes, and the forest breathes. It remembers an age before scissors could cut life's script, when evolution was slow, messy, tangled, and perhaps it was kinder for being so, because it left room for mistakes, for surprise, for the kind of life that grows sideways before it finds its shape. You open your eyes and ask, is the wild still wild when we can reprogram its deepest code? Or has it become a garden we think we tend? You walk on now toward the ocean's edge. Here, a coral reef glows with tentative hope, its polyps edited to withstand rising heat. CRISPR, once again, as savior. And yet, the reef sighs, because the real heat comes not from genes, but from choices, from industries, from consumption, from the slow burn of convenience. Edits to coral by time, but time without action is not salvation. You listen to the waves and realize the wild may accept our changes, but it will also reflect our values, not just what we can do, but what we choose. You sit now on a stone, the stars above, whispering slowly across the sky. A fox watches you. Its eyes are clear, unafraid. Within it, a virus has been edited out. No rabies, no fear. And yet, it is still wild, still free. You smile, because CRISPR is not inherently unnatural. In careful hands, it can be a bridge back to balance, not to conquer nature, but to accompany it, to restore what we once broke, to heal with humility, but only if we walk slowly, only if we remember that every gene is part of a story longer than our lifetimes. And so you rise, you brush the dust from your palms, you listen to the wind, to the animals, to the hush between the edits, because the next chapter does not unfold in labs or forests, but in cities, in policies, in power, where decisions about the wild are made in boardrooms. And so, gently, you walk now from the woods into the world of law, where CRISPR faces the question, who gets to control the code of life and who gets left behind? You lie still tonight beneath a sky that flickers not only with stars, but with possibilities. The future feels close, not distant, not a far horizon, but something humming quietly just beneath your skin. The air holds questions. The wind smells like change. You breathe in and begin to see it, not as a single road, but as a patchwork, not smooth, not seamless, but stitched, square by square, by the choices we make now, a quilt of tomorrows, some soft, some frayed, some dazzling. Each square, a different vision of what life could become. Each thread, a hope, a warning, a dream. You walk gently through this imagined fabric. You see a city square, not grand, but quiet, clean, bathed in morning light. Inside a small hospital, a baby sleeps peacefully, her genes edited to remove a disease her mother once feared. She will never know the word sickle. She will never feel the ache in her bones when winter comes. Here, CRISPR is part of care. Safe, tested, reviewed by ethicists, approved by law, guided by kindness. You pause. There is peace here, but the next square is not the same. You take one step, and the scene changes. Another hospital, another mother, another child. But here, there is no edit. There is only waiting. There is only hope and paperwork that never moves fast enough. The procedure exists. The science is known, but the policies lag behind. And the cost is everything. In this square, CRISPR is a distant promise. Too far, too expensive, too political. You feel the unevenness in your feet. The patchwork shifts. You begin to see that the future is not fair unless we make it so. Some nations leap forward. Others hesitate. Some restrict. Some exploit. In some hands, CRISPR is a public good. In others, a private investment. You move now into a brighter square. A classroom. Children giggle. They trace molecules in the air with fingers stained by art, not ink. One child solves equations with ease. Another composes music in silence. You wonder how many of these gifts were shaped before birth? How many came from genetic nudges? Tiny, silent edits made with love or ambition, and suddenly a gentle unease rises. Not every edit is for healing. Some are for height, for speed, for attention span, for excellence. But when does striving for more cross into rewriting what it means to be human? You picture a world where only some get to optimize, where wealth buys advantage, not just in schooling, but in biology, where the word natural starts to fade from the tongue. The patchwork shivers. You walk again, this time into a field, small farm, dusted with golden light. The crops here are strong, resistant, kind to the soil, not because they were sprayed, but because their seeds were edited to adapt. You meet a farmer. She smiles, weather-worn and proud. We saved the beans. She says, the blight came, but this time we were ready. CRISPR, used here, is quiet and local. It is stewardship, and the seeds shared freely with nearby villages. Another square, a rainforest, reborn. The trees grow fast again. The fungal disease that once stripped their bark no longer wins. The birds have returned. The jaguar tracks are fresh. Science, woven with conservation, has done more than just rescue a species. It has restored an entire rhythm. You pause again. Because this is not about edits, it is about intentions. The same tool that divides can also mend. You begin to understand. The patchwork future is not one future, it is many. Some stitched with care, others sewn in haste. And the quilt we sleep beneath will depend on who holds the needle. You float upward now, above it all, above cities and clinics, forests and labs. You see the quilt in full. It stretches across nations, across cultures, across centuries still to come. It is dazzling, not because it matches, but because it doesn't, because it reflects our complexity, our beauty, our contradiction. It holds mistakes and miracles, and in every square, a question. What kind of ancestors will we choose to be? You drift down into a final square. It is not yet stitched. It waits for your voice, your care, your caution. Because even if you never edit a gene, you shape the future with your values, with your vote, your attention, your conversations at dinner, your questions in class. The patchwork is not a product, it is a process. And every voice adds a thread. You lie back now. The quilt folds over you, not to smother, but to cradle, not to control, but to warm. And you feel it, this strange comfort that the future is unknown, but not unwritten. It is being sown, even now, in whispers, in labs, in lullabies. The patchwork breathes. And just before you drift to sleep, you feel the hush of the next chapter coming, a chapter not just about science, but about time, about what we leave behind, about the stories we tell with genes and memory, about how we pass on more than blood. We pass on choice. We pass on care. We pass on the responsibility of the edit. You lie still, cradled in the hush of nearly dream. Around you, the patchwork future still folds softly, stitched from human hands, stitched from questions. But now you turn inward. You look at the tool itself, at CRISPR, at the editor, and you ask, what happens when the editor begins to edit itself? The night feels recursive now. A mirror inside a mirror. A spiral of thought, curving gently inward. You drift to the edge of a laboratory, lit not with bright light, but a soft, flickering glow, like candlelight made of code. Here, scientists sit quietly. They are not programming machines. They are programming the editors that once edited cells. Layer upon layer, the work has grown. What began as a borrowed bacterial trick, a tiny pair of molecular scissors, is no longer just scissors. It has become a system, smarter, faster, more precise. You watch as CRISPR is tuned by its own kind, versions of itself built to be better. Some can find a single mutation in a sea of code. Some can change a single letter without cutting the strand. Some turn genes off, like dimming the lights, without ever breaking the glass. You walk slowly along this hall of mirrors, and you wonder, what are we teaching the editor to become? Not a tool, but a partner. Not passive, but almost thinking. A whisper drifts to you. Base editors. Prime editors, tools inside tools, like nested dreams. Each new layer not only fixes more, but begins to choose what to fix. Not only cuts, but decides where and why. You drift now into another square of the future, a clinic without surgeons, only molecules at work, editing in real time, guided by algorithms trained on human history. A child's blood carries a message to the editor, and the editor answers without noise, without incision. A single typo is corrected before the child even knows what it might have meant. It is beautiful. It is delicate. It is invisible. And yet, you feel a tremble in the thread because something new is unfolding here. You look at your hands, at your breath, at your cells, and you ask, if we can edit the editor, where does the editing end? You float into the memory of early tools. A hammer, a wheel, a loom. Each invention changed what we could do. But this tool changes what we are. Because if CRISPR learns to improve itself, to design newer forms of itself in loops of recursion, then it begins to walk the path of evolution. Not natural, but artificial. Not random, but refined. And somewhere in that loop of editing the editor, of building the builder, we lose the sense of beginning and end. You hear a voice now, a soft one, perhaps your own. It says, tools were once our extension. Now they begin to extend themselves. The voice does not judge. It only observes. And you feel it, the weight of this recursion. Not fear, not joy, but you walk further into a lab where CRISPR writes new proteins from scratch, not borrowed from bacteria, not stolen from nature, but dreamed, modeled, simulated, printed in atomic ink. These editors have never lived. They were not born. They were designed. And yet, they live now in cells, making changes, writing futures. The editor has become a language, and we are teaching it how to speak. You move now into a slower room, a circle again. Bioethicists, artists, students, farmers, parents. They are asking, if we allow the editor to evolve, who holds the leash? And if it improves itself beyond our grasp, do we still hold responsibility? Or does the line blur between creation and creator? You rest your eyes and picture a painting unfinished, a canvas where each stroke paints the next hand to hold the brush, a recursive masterpiece. You wonder, can we build responsibility into the spiral? Can humility be coded into a tool? Can we teach a molecule to remember the weight of choice? You float again into a garden, the same garden from before. But now, the plants repair themselves, not just immune to blight, but aware of how to adapt. The code that edits them lives inside their cells, listening. You see life not just surviving, but rewriting its own resilience. And again, you wonder, have we crossed some unseen line? Or have we simply moved deeper into the heart of nature's own intention? Because evolution has always been An editor. Slow, blind, patient. Now, it has a guide. You lie still once more and feel the gentle pulse of recursion. You do not fear it. You do not fully understand it. But you know, it is here. CRISPR is no longer just a tool. It is a language, a process, a mirror. And the moment it edits itself, we must ask, what values did we place in that first reflection? What stories will be carried into every self-edited editor to come? Because every recursive tool holds its origin like a seed, and that origin is us. You exhale slowly. The lab dims. The mirrors soften, and you drift gently toward the final chapter, not of CRISPR, but of our relationship with it. Chapter where the question is not what we can do with the editor, but what we choose to remember about being human as we place that final stitch in the patchwork, the echo of a breath, the imprint of choice, the humility of hands. You feel the silence bloom, and the final question lingers. What happens when the story we write begins to write us back? You lie beneath the hush of everything you've heard. 17 chapters now, folded like petals around you. Each one a breath, a step, a spark. And now you arrive at the final one. But this chapter is different. This chapter isn't about CRISPR, not entirely. This one is about you. Because in all these stories of genomes edited, of futures shaped, of ethical mirrors and spiraling code, the quiet truth was always waiting. You are not separate from this. You are not only the listener. You are the unwritten chapter. You carry within you 3 billion letters, whispering through every cell, a library made of light and dust, written over eons, and still not done. Your body is a quilt of inherited stories, some ancient, some brand new, stitched in double helix thread. You are not just the sum of your parts. You are a story being told in real time. And science, this quiet, curious unfolding, has merely held up the mirror. You think back now to the bacterial guardian fighting viral ghosts, to the scalpel of light slicing with impossible grace, to the cry of the first CRISPR baby and the ethics wrapped in helices, to the ghosts of eugenics, the garden of John pools, the recursive editors that shape themselves. You remember the whispered questions left behind in each room. Should we? Who decides? Where does it lead? And now you feel those questions turn inward, because here, at the end, they belong to you, not to labs or governments or molecular tools, but to the person you are becoming every second. The edits that shape the world now are not all molecular. They are choices, moments, small kindnesses that ripple through generations like soft echoes in the code. You are made of decisions your ancestors never imagined. You are made of resilience your cells don't even know they carry. And as the editors grow smarter, faster, more recursive, you remain the most unknowable element of all. You, the unpredictable, the dreamer, the poet made of carbon and care. And so you rest now, not with certainty, but with permission. permission to wonder, permission to not know, permission to grow slowly in a world racing toward precision. Because the chapter called You cannot be mapped on a genome chart. It is written in laughter, in sorrow, in silence, in the gentle act of asking questions you may never answer, and yet that asking matters. In fact, it may be the most human gene of all. You remember the mobile, spinning above the crib of our future. Each molecule, a star. Each choice, comet. And in the center, stillness. A child, not yet named, not yet edited, simply becoming. You may never edit a gene. You may never hold a pipette or open a DNA sequence on your screen. But you will love someone. You will teach someone. You will ask them what they believe. And in that, you change the future as surely as any CRISPR strand. Because science does not only live in petri dishes, it lives in kitchens, in hospitals, in bedtime stories, in the quiet act of choosing empathy over ease. So you breathe in now, a slow, warm breath, and you feel your own cells stir, billions of them, working, healing, whispering. Your code isn't perfect, but it is yours, and it is still unfolding. You are not a machine. You are not a product. You are a story, being told in moonlight, in uncertainty, in hope, and you are not done. You never were, because the chapter called You does not end here. It does not end at all. It lives in the hands you'll hold, the memories you'll shape, the questions you'll leave behind. And maybe, one day, someone will find your name on a list of those who paused, who listened, who dared to ask softly, what kind of future feels like home? And so you drift now into that future, not with fear, not with urgency, but with a quiet kind of bravery, the kind that holds both caution and curiosity in the same breath, the kind that remembers you are not the editor's master, you are not its subject, you are its companion. You walk beside it, and tonight, as sleep rises like a tide, you let go of the noise. You return to your breath, to your cells, to your story, unfinished, unfolding, glowing softly from within. Because the last chapter was never in the book. It was always in you. Good night, unwritten one. Your code is waiting. Your questions are sacred, and your presence is enough. And now, dear dreamer, let your mind rest gently, like stardust settling across the night sky. Whatever questions still linger, they can wait for another evening. You're part of this story, curious, soft, and human. Thank you for wandering with me tonight. This has been the Dreamy Physicist, a quiet journey through the origins of time. Until next time, breathe easy, sleep well, and let your thoughts drift like photons through space. Good night.